This procedure begins with fixing the bee into a recording chamber. A piece of cuticle is removed from the head capsule to stain selected structures in the brain. Calcium signals in the stained neurons are recorded in vivo using a wide-field imaging setup. During imaging, the bee can be stimulated with odors or sucrose delivery to the antenna. This way, the bee may be conditioned to extend the proboscis in response to an odor. The proboscis extension response is monitored using electromyogram recordings from the M17 muscle. After the imaging experiment, the brain is dissected for a closer investigation of the stained structures using a confocal microscope.
Hi, I'm Melanie from the lab of Randolf Menzel.
Hi, I'm Anja, also from the Menzel lab.
Today we will show you a procedure for in vivo calcium imaging in the honeybee mushroom body.
We use this procedure in our laboratory to study olfactory coding and learning-related plasticity in the honeybee brain.
So, let's get started.
Start by catching a honeybee forager at the hive entrance. And then immobilize it by chilling it on ice. Next, mount the bee onto a plexiglass recording chamber